Thank you all for joining us, and our deepest appreciation to Keystone for consistently convening such a singular forum and allowing us to celebrate the bold vision and reach embodied in the research represented here tonight and across the many conversations this week. My name is Philip Kim, Executive Director at Michelson Philanthropies. It's my privilege to welcome you to this evening's recognition of the recipients of the 2026 Michelson Next Generation Grants. Just a bit of context. For over really 3 decades now, our organization has quietly funded various medical research projects across the spectrum and all towards the creation of a healthier and more compassionate world and a North Star of ending disease as we know it in our lifetime. And in this moment where science and science funding in the US are under unprecedented threat, we are more energized than ever. Between just our active Center for Convergent Bioscience at the University of Southern California and our new partnership with UCLA to launch the California Institute for Immunology and Immunotherapy, we have committed just under $200 million towards the discovery of novel life-saving technologies. And to directly meet the challenges of this moment, Dr. Michelson is also spearheading a new California ballot measure in November to mobilize $8.4 billion towards immunology research and cures across the state's research hubs. But the reality is we see all such projects and all of our work with governors and congressmen and NIH leadership to strengthen the infrastructure and culture and the innovation ecosystem really as secondary. It's all intended to cultivate the conditions within which bold scientific vision and individual brilliance like that we are here to recognize tonight can flourish. And in particular, the Michelson Next Gen Grants support the daring work of early career researchers navigating the limitations of traditional funding pathways. And they provide a platform to catalyze broader investment and accelerate both science and careers. We've asked Dr. Michelson to share a bit more on the spirit of this program now.
More than ever, we need disruptive thinkers and doers who will change the trajectory of medical science for the better. I am Dr. Gary Michelson, the founder and co-chair of Michelson Medical Research Foundation. It is beyond difficult for young scientists to get funding to conduct their own research and to do their own out-of-the-box, potentially high-risk, high-return work that would disrupt the status quo. The Michelson Prize's Next Generation Grants are changing this. We support outstanding early career investigators who want to perform disruptive research and are working to advance human immunology, vaccine development, and immunotherapy discoveries across major global diseases. When we empower young, brilliant minds and invest in high-quality, cutting-edge medical research, it's with the idea that a single scientific breakthrough discovery could affect the lives of hundreds of millions of people. So be bold, go big, disrupt the status quo, accelerate your career path, and apply for the Michelson Prize's Next Generation Grants. Thank you.
So the collection of early career innovators with us tonight, who will each be receiving $150,000 to advance their work, represents our largest cohort in the decade of the program. So we would like to dive right in. And before we hear from each of them, I'd also like to recognize Lisa Wagar, a 2022 recipient of the Michelson Philanthropies and Science Prize for Immunology, who was honored for her pioneering work in immune responses to viruses and vaccines using human tonsil organoids. And we're delighted to have her here with us this evening. Lisa, if you're here, thank you for joining us and for being here again. Okay, first I'd like to introduce and bring up Dr. Mohammed Abedi, currently an HHMI Fellow in David Baker's lab at the University of Washington. His work focuses on engineering immune therapies that activate only as needed, such as within tumors, thereby minimizing off-target toxicity and enhancing both safety and precision. This summer, we are also thrilled to welcome him back to California as he launches his own lab at UCLA. Please join me in welcoming Dr. Abedi.
Okay, hello everyone, and I think this is the hardest presentation that I had to give in my career so far because I'm competing with food. And I'm a person who loves food, and while you enjoy the food that's going to feed your stomachs, I'll try to give you some food for thought. And I promise that that's going to be exciting, right? Like I thought about this, I was like, how do I get people to pay attention? And I structured the presentation in a way that it's general, but I think it will kind of get you excited about the area that I have been looking into, and in the future my lab is going to be looking into. And basically it really boils down into that we as humans, we really care about communication, right? And we were able to build this like, amazing societies, amazing civilizations. You can go around Ireland and see a lot of it. And the key there is that we were able to communicate between each other to build bigger things, right? Like something that is bigger than the individual. And it turns out that the key of the cells that are within us to do the same thing, right? Like for a single cell to build an organism, really at the center of it is communication, because these cells have to talk to each other. But they don't use words, right? Like when we talk, we use words, we vibrate air, and somehow me vibrating air right now makes complete sense to you and you can understand all these complex ideas. But cells don't do that, right? Like so cells exchange proteins and metabolites. And something that like, it's interesting for me to think about is that this communication is really central to biology at many different levels, right? Like development. Cells have to talk to each other to push a program, and it has to be perfect. You cannot really have any errors. Another area is in the tumor environment where there's a lot of interactions between your cells, your immune cells, the native tissue. And finally, this communication even happens between species. So plants and microbes in the soil talk to each other all the time. What I'm going to focus on today is communication that happens specifically in the tumor environment. So it's going to be localized there. And many of you have talked about this, and as people who think about vaccines and the immune system, you know that cytokines are one of the main molecules that the immune system uses to communicate between each other and with other cells. And for me, fundamentally, what a cytokine does is that it just brings two receptors together, right? Like it's a very simple mechanistic thing that needs to happen on the surface of a cell. You bring two receptors together. There's a lot of things in the middle that happens that honestly, a lot of it we still don't fully understand. But at the end, it tells the cell to do something specific, right? And when we take the communication analogy again, when we talk, we use words. And to make words, we assemble letters together. And it turns out that certain combinations of letters are much more useful to form words than others, right? So this is frequency of letter usage analysis that was done in the 7th century, right? So we knew this for a while, that not all combinations of letters are as useful for speaking. So like ZZ, for instance, you don't see that much in words. And it turns out that for cells, for clustering receptors, the same is kind of like you can kind of see it, right? Like there's a lot of receptors on the surface of a cell, but there's only a limited subset of cytokines. And if you want to put numbers on it, average cell will have about 100 unique receptors. And these receptors, if you want to cluster all of them in all combinations, 100 times 100 is 10,000, but the unique ones is half of that, so it's 5,000. But we definitely don't have 5,000 cytokines, right? And when I started thinking about this, I had the question of like, why? Like, why did nature not make all the 5,000 different cytokines, and why did it stop at about 100 or so that we know and we use all the time now? And before, there was nothing we can do about it, right? Because the way we study biology is we go look at what nature does and try to understand the proteins that exist in nature. The really exciting thing is that recently there has been this technological breakthrough, computational protein design, that allows us to build proteins from the bottom up, right? So we don't really care if nature has made a protein or it didn't. We can make the protein that we want from scratch. And equally important, like it's really cool to make proteins on a computer screen, but you need to take that into the real world, and you need to set up assays so you can ask, like, what happens when a cell sees these like new proteins that we are building? And that's exactly what we did, right? And the first step is that to build a cytokine, what you really need is a binder that interacts with these receptors. And once you build that binder, you fuse two binders together, and that induces clustering, and that's basically it. And technologically, what that looks like is that I go on a computer, I design a binder. Here I'm showing interferon receptor 2, and I'm showing the binding affinity of it. And it's pretty simple at this stage. So as a lot of you know, that making a binder is not that much challenging. So we made a lot of them, and we made about 33 binders. And then we went to the lab. And when we first started to make 48 proteins, 3 of us, it took us 3 days. And we knew that building new cytokines that can reach the 5,000 cytokines that nature didn't make is going to be really hard. So what we ended up having to do is to build this like high throughput system where we can express these and test them on cells. And then I can either relax or I can focus on more important things. But now that we can express, we can design all these new cytokines that cluster new receptor pairs, and then we can test them on cells. We took PBMCs and we asked what's going to happen, right? And similar to that matrix that I showed you of letters, now we have this matrix of receptors that are dimerizing with each other. And here I'm showing STAT signaling. So we only looked at the STAT signaling pathways, STAT 1, 2, 3, and 5, and 6. And the exciting thing here is that a lot of it was signaling, right? And just to give you a perspective, over here, only 5 are natural cytokines. And everything else that you're seeing is, as far as we know, since the first mammal, which is Murogon, we haven't heard, right? Like these are cytokines that are signaling in our cells and mammalian cells for the first time, and we had no clue what they are doing. So we wanted to check, like, okay, is it just signaling in receptor pathways, or is it actually inducing function? And one of the things that we found very exciting is that one of these cytokines, which dimerizes interferon 1 with IL-7, drives proliferation in T cells just as well as IL-2. And I would not have guessed that. Like, why would IL-7 and interferon 1 be as efficient as driving proliferation as IL-2? We don't know that. And just to give you like an expansion of what is possible now that we are expanding the vocabulary of cells, it doesn't really stop at immune cells. So we went to here stem cells, and we asked if we use these new words to communicate with the stem cells and try to drive them into muscle cells, can we enhance that process? And what I'm showing at the bottom is that there's 2 combinations, IGFR and PGF, and EGFR, which the names don't matter, but basically what's happening is that we're finding these new words or these new cytokines that allow us to communicate with stem cells and enhance the efficiency of differentiation. So this really goes beyond the immune system. It's just we're communicating with cells in general in these new ways that we barely understand. And it doesn't really stop there, because another thing that we realized is that we don't only make new words, but we can also change the tone of the word. And we do that by changing the geometry of clustering, by just choosing what internal sides, what the domains on the inside of the cytokine are facing. And when we do that, so we tested this with type 1 interferon, and you can see that we can modulate the activity of the STAT1 pathway. And why does that matter? Because when you induced a cell with a cytokine, a lot of things happen. So in the case of interferon, you get upregulation of MHC1 and of PDL1. And as you know, these are things that do the opposite thing, right? Like one is great for stimulating the immune system and the other is not. And what we wanted to do is to decouple them. And by modulating the geometry of cytokines, we were able to downregulate PDL1 but upregulate MHC1. So you can really use the same receptor pair but modulate what gets activated. And here, really, the vision is that we're going from cytokines towards these de novo designed cytokines, which we call novokines, which allows us to, 1, expand the language that cells use, and 2, bias signaling and kind of change the tone of how these cytokines talk to cells. And finally, what the Michelson Foundation is supporting us to do is to build new words that specifically talk to a cell that we want to target or in an environment that we want to target, because words that are more targeted are useful as opposed to things that you broadcast to everybody that could cause toxicity. And we're going to do this work at UCLA, where I'm starting my lab, so I'm very excited to push these directions with the support of the Michelson Foundation. And there we're going to ask, like, how can we bring tools from protein design and synthetic biology to go beyond what is available in nature. And with that, I would like to thank the people I've worked with. I'm adding here links to the preprints of some of the work that I described. And in addition to making high-throughput cytokines, me and Mark have optimized a protocol to make high-throughput falafel. So if you're interested in eating more beyond what you're having of delicious food today, ask me and I'll give you the protocols for that part. And thank you very much for listening.
Thank you, Dr. Abedi. Next, it's my pleasure to introduce Dr. Theodore Roth, an MD, PhD, and assistant professor at Stanford University. His research ventures beyond the limits of natural biology, re-engineering human immune cells in fundamentally new ways to generate more potent and effective responses against cancer. Please join me in welcoming Dr. Roth.
Hey everyone, my name's Theo Roth. I'm a new faculty member at Stanford University. And while you're eating, I'm going to try to get you to imagine what immune cells could do that they didn't naturally evolve to do. And as for a vaccine conference, we try to get immune cells to do things that they weren't doing naturally already. We try to get them to be specific for diseases that we've never seen. So hopefully you guys are a receptive audience that immune cells can do things that maybe nature hasn't evolved yet. At the core of how I think about a lot of this stuff is that DNA encodes for everything. So if you can engineer DNA, you should be able to engineer cells to do whatever you want. That's vaguely what evolution does. So within the context of immune therapies, across many different classes, we're trying to take the existing specificity of the TCR and BCR repertoire and the existing cell states and functions that they have, and then alter that, whether that's through indirect genetic engineering, such as giving a vaccine, or whether that's through direct genetic engineering, like going in and actually editing the DNA of a human T cell or a human B cell to just give it the TCR that you want or give it the BCR that you want. And so my work for about a decade now has focused on generating ever more powerful tools to insert new DNA into human immune cells. And so if you want to take a number away from any of this while you're chewing on stuff, we can put well over 10,000 base pairs into the genome of primary human T cells, and we can test them in batches of about 20 to 30,000 sequences at a time, and that's on a weekly basis. So there's massive power to put new genetic material into human cells and directly assay what it does. And so the toolkit that we have for genetic manipulation of immune cells has drastically expanded. So if you're not in this field, please let me tell you, you can do incredible things that were not possible just 2 or 3 years ago. And so many of you may have done CRISPR screens or used single genetic perturbations, ORFs, to overexpress something or put in one TCR or one BCR to make a genetically engineered clone. We can go well beyond that now. We have a tool that's up on bioRxiv called CRISPR-All that allows you to do programmatic encoding of genetic perturbations into the genome. And so it's essentially a programming language that you can dial in between 4 to 10 genetic perturbations arbitrarily across different perturbation classes, and you get a human immune cell that has those 4 to 10 perturbations back. And you can test all of this simultaneously in large-scale pooled screens. So think about what immune cell you like to work with or what you want to alter its function. Think about all of the knowledge that you've gained from all of your training and say, what are the genes that you want up? What are the genes you want down? What are the genes you want out to get that cell to do the thing that you've always wanted that DC to do, always wanted that B cell to do? We have a tool that we think you can explore that and then your other 10,000 hypotheses as well, simultaneously in a pooled setting. But you might say that, what if there is no combination of natural genes? The genome has 20,000 levers in it that you can pull up, down. And Mohammed gave a great example of how you can go beyond the natural alphabet, the natural vocabulary of cytokines. And we think of it in a very similar way. What happens when you go beyond the natural set of genes? And you just think about it, many of us study model organisms. You don't have to go that far back in evolutionary time where our immune system did not have 20,000 genes to work with. It only had 16,000. It only had 8,000. You go back long enough, it only had 3,000 genes at the last universal common ancestor. So we've evolved new genes in the past to give new functions. So the next question to think about while you're moving on to dessert is, if you could give an immune cell a new gene to optimize its function for the setting and behavior that you want, could you ever find something that would be useful? Or do you think the 20,000 that we've got cover everything that you could possibly ever want an immune cell to do? I'm a pathologist. I see dead people in my clinical practice. I can tell you the immune system does not do everything we want it to do. And so the next generation of what we've been focusing on, what the Michelson Foundation has supported, is exploring whether you can actually use these new genetic perturbation tools. Now that we can test enough sequences and enough throughput, can we actually find new genes that didn't naturally evolve that can give immune cells new functions that can better fight disease, better fight tumors. And so with that, I'll just leave it and say that we've done this in T cells. We've actually found a lot of them. We've got a couple of preprints up if you want to check out our lab. But we can do this across the hematopoietic lineage. If you want to explore high throughput genetic questions around what is the next step in my favorite immune cell's evolutionary trajectory to cure that disease, to have that functionality that we've always been trying to get, please reach out. We would love to see these systems applied in as diverse systems as possible. My email is on here. You look up online. We love to get genetic engineering tools into the hands of the experts that know what questions to ask using them. So please reach out. We'd love to talk to you. And thank you to the Michelson Foundation for supporting the most ambitious stuff that we can imagine doing. So thank you.
Thank you, Dr. Roth. Okay, moving right into our next speaker, Dr. Inta Gribonica is an assistant professor at Lund University, and her work explores how beneficial bacteria on our skin can enhance immune protection, opening the door to new topical therapies and innovative vaccine strategies that strengthen the body's first line of defense.
Good evening, everyone. First and foremost, I want to thank the Michelson Medical Research Foundation for this opportunity to talk to you. I would like to share my story about skin humoral immunity and how can that be repurposed for alternative vaccination strategies in the light of this wonderful conference. So let me start by saying that mucosal tissues and the skin collectively form the outermost surface of the human body to the external environment. And as such, these tissues are extremely prone to injury, caused by a variety of factors such as dietary change, aging, partnership, travel, insect bite, and many others. So when the tissue is compromised, this brings the opportunity for pathogens to cross our natural borderline with the outside world by causing infection that eventually can develop into serious disease. So the best way to fight off such insults, of course, is very strong tissue immunity that can be caused by boosted sort of local vaccination responses targeting resident T cells and B cells, and therefore leading to antibody production as the first line of immune defense. And we all know that within gastrointestinal tract, which is the largest mucosal surface, we have a very interesting and very effective way of doing so, because every single external stimuli, including that of microbiota, triggers continuous activation of immune cascade via germinal center reaction, leading to high affinity antibody production in mucosa. And this is really the baseline for how we think about oral vaccine design, right? But when it comes to the skin, the situation is a little bit different. You see, the state of skin immunity says that consensus would be that skin actually is completely devoid of B cells. But is that really true? You see, more and more clinical cases show that many skin diseases actually are characterized by B-cell infiltrates with context-dependent functions. And even at homeostasis, up to 70% of healthy human volunteers have very high serum reactivity to skin-homing microbes without any prior infection whatsoever. And even more, if we take human skin swabs from healthy individuals, we can actually extract commensal microbiota labeled by antibodies. So with all this body of literature, I formulated my research question, and that is whether skin colonizing microbiota actually can stimulate antibody responses. And to do so, I utilized a very well-established model system in our laboratory, that is Staphylococcus epidermidis topical association, which literally means that you paint bacterial culture on the surface of the skin for 4 consecutive days and let mice to rest. And as you do so, you actually develop very strong association between the microbe and its host. And this is a perfect model to ask this question, whether through such association you are going to stimulate antibody responses systemically and locally. And in fact, yes, 30 days after association, you have very strong IgG titers to skin colonizing microbiota without any form of infection. And this response, in fact, is modulated through germinal center reaction, requires TFH cells, and even more so, Langerhans cells. Okay, so Staphylococcus epidermidis without inflammation can stimulate classical GC responses, as we know in lymph nodes. So do we find these GCs? Yes, we do. Here you have cross-section of skin-draining lymph node before and after association, and you see with this green B cell 6 area that ongoing germinal center responses induced. But, I mean, we are putting bacteria on the surface of the skin, right? So something must have happened in the tissue itself. So, to investigate this question further, I looked into eardermis hormones, and I hope you can appreciate that association event itself completely reorganized skin tissue by inducing large dermal T lymphoid structures that were characterized of B cells and T cells, and both of these expressed BCL6, indicating of local skin germinal center activity. So when I look at these structures, what I find is that these are forming around hair follicles. And I think that hair follicle in this case acts as nature's natural needle for allowing bacterial antigens to penetrate deeper into the tissue and to be accessible to surrounding immune cells. So now this paper, this study is published, and I'm not going to go into detail as to how it's been done and how it's going to happen. But what I want to say is that what we have identified is that skin is not devoid of B cells. In fact, at steady state, you have quite a bit of B cells that are very much surveying microbiota, reacting in response to colonization, forming germinal center responses at steady state without inflammation. And this structure, TLOs, these are very, very important for maintaining barrier integrity of skin tissue, okay? And one more thing. These are very much independent of the draining lymph node germinal centers. That's also a very interesting point. So how can we take this observation or discovery further for the benefit of patients and benefit for the society at large? So my collaborative work with Janet Bose-Bain and Michael Fishback from Stanford University has already shown that if we engineer Staphylococcus epidermidis to express pathogen fragments on its surface, and then you utilize it for colonization of the host, you not only induce bacteria-specific antibody responses, but you're also inducing very protective immunity against the pathogen itself, both systemically, at mucosal surfaces, and locally within skin tissue, right? And this is the work that probably Janet and Michael is going to follow up very nicely. But what I want to do with the support of Michelson Foundation is really to think about wild-type microbiota-induced tissue infrastructure and how that can be repurposed for vaccine delivery within cutaneous compartment in more time-restricted manner, limiting any negative long-term adverse events from the fact that vaccinal modality could stay with us for a very long time in the first setting. So let me just give you one last data slide to show that this could actually be feasible. So what I have done, I have associated mice with Staph epi, and 90 days later, I checked whether there are dermal T lymphoid structures induced, just using wild-type microbiota. And then I transfer over specific T cells and use our favorite over immunization model by basically topically applying ovalbumin on the surface of the skin in the area where TLOs are present. So what I then see 45 days later, that in fact on the skin surface, you actually have over-specific antibodies if the TLOs have been there through prior associations with Staphylococcus epidermidis. This leads to the situation that your skin is not only labeled with microbiota-specific antibodies, but in fact, it also has a shield of vaccine-specific antibodies that can be your first line of immune defense upon a pathogen attack. So to take this interesting observation further, I want to explore how TLOs can be induced in a more controlled way in the context of sexual dimorphism, because there is huge difference between males and females, and across the lifetime, basically, looking from the young individuals to the old. And the second very important aspect that I want to further explore with this grant is basically to understand what is the quality and kinetic of tissue-resident skin B cells, and how they relate to systemic counterparts, both in situ localization in mouse and human tissues. So what is the long-term impact of this type of work? Well, first of all, I already spoke, this is vaccination, and I think if we can take this further, it could be a nice addition to our attempts to create mucosal vaccine. A topical vaccine would be basically formulated in creams that you can actually apply yourselves at homes, if it works. Then there are other parts that are very important. And topical association and microbiota-induced immune responses, especially B cells, they are very much telling us about how chronic skin inflammatory diseases might occur. Because many chronic skin diseases, such as hidradenitis suppurativa, actually are characterized by B-cell infiltrates, microbiota dysbiosis, sexual dimorphism, and so on and so forth. So I think it would be very interesting to see how this could pan out and how we can actually understand better strategies for skin disease treatments. And finally, if we know how to induce tight lymphoid structures or how to switch them off, we could actually benefit patients who are either undergoing gene therapy or cancer immunotherapy. So all in all, I think there is huge potential in skin B cells, and therefore I'm extremely thankful to Dr. Michelson, the Michelson Medical Research Foundation, and Next Frontier Advisors for this very special recognition for allowing me to continue this type of work in my lab. I also want to thank my scientific advisors over the past 12 years, Dr. Professor Nils Lycke and Yasmin Belkaid. And since about a year ago, I've started my own lab at Lund University in Sweden. So I want to say thank you to my current lab members for their current work and the future work that's going to be done with this grant. So if you have any questions, I have poster tonight, so please come by and let's talk. Thank you so much.
Thank you, Inta. Last but not least, Dr. Joshua Gray is an associate research scientist at Columbia University, and he examines how infant immune cells interact within tissues such as lungs and the gut with the goal of designing vaccines to provide stronger, more durable protection from the earliest stages of life.
Hello, everyone. So I'm going to talk a little bit about a little bit of data on how CD4 T cells are imprinted by the tissue in which they reside. So we kind of all know that CD4 T cells have tissue-specific functions based on where they are in the body. And the best example of this in terms of effector function in a lymphoid tissue is follicular helper T cells, as we all know and as we've all discussed throughout the rest of the conference previously. And these TFH cells, they're characterized primarily by expression of PD-1 and CXCR5, which allows them to move into the germinal center of lymph nodes where they express IL-21 and essentially help B cells produce antibodies and provide protection to an infection. But in mucosal tissues, it's not the same as that. So we have TRM in mucosal tissues, which express various markers that allow them to be retained in the tissues, such as CD69, CD103. And they also have enhanced effector functions that allow them to provide rapid protection to a pathogen in which they're encountered, such as interferon gamma, granzyme B, TNF alpha, things like that. And my kind of interest here is that we're not born this way. We don't generate we take years to generate these sort of T cell responses and this tissue-specific CD4 T cell response throughout the body. So that was the kind of question that I approached when I applied for this grant. And to do this, we have a couple of resources in the lab that allow us to really interrogate this in humans. And the first one is this collaboration that we have with Live On New York, where we get adult organ donors from various tissues throughout the body, including lungs, gut, liver, spleen, things like that. In addition to that, when I joined the lab, we set up a new collaboration with various organ procurement organizations throughout the US that allowed us to collect tissues from otherwise kind of ostensibly healthy pediatric organ donors. And I know that's kind of an unpleasant thought, but it allows us to collect these tissues that otherwise can't be used for life-saving transplantation. And what this does is it allows us to kind of interrogate live cells in their native tissue context over the course of a range of ages that allows us to build up a picture of how these cells develop in a tissue-specific manner. And one of the main things that we found when we kind of when we did this, we published this a couple of years ago, is that in the lungs of children, they have these clusters of T cells and B cells that allow them to produce antibodies locally at the site in which they're kind of bound to encounter antigen. And these bronchus-associated lymphoid tissues, they're able to produce antibodies that are specific for respiratory viruses like RSV and influenza. But the kind of take home here, the most important thing about this is that they're very transient. They go away. They take a couple of years to develop, and by the age of 5 or 6, they're fairly undetectable, and we've never really seen them in adult human lungs. And kind of my approach here, this kind of paper was kind of focused on the B cell side of things, but I'm a T cell immunologist, so I could kind of come in here and ask some fairly basic questions about what's going on in these bolt structures, and are they operating on the same paradigm that lymphoid follicles are in the kind of paired draining lymph node. And to answer this question, we took advantage of our pediatric organ donor resource by looking at paired lung and lung lymph node samples. And the fact these organs are from the same donor, that accounts for a lot of the high variability we've talked about, characteristic of human organs. And kind of one of the first bits of information that we found was that the lungs completely lack one of the lineage-defining markers, CXCR5, that's characteristic of TFH. Even though we know that these bolt structures can produce antibodies, the lungs lack the actual cell type that we know is essential for B-cell help. So we took a deeper dive into these cells by doing some transcriptional analysis on some memory CD4 T cells, and I wanted just to focus on clusters 3 and cluster 8 here. Cluster 3 is your classic TFH signature, gene signature, whereas cluster 8, that's kind of a signature that doesn't really fit with any of the sort of well-known helper CD4 T cell subsets, and it's characterized by expression of multiple functional molecules such as PRDM1, interferon gamma, IL-21 importantly, as that's the cytokine that's used to help B cells, and IL-10 as well. And when we stratify these cells by tissue, you can see that the TFH are specifically in the lymph node, whereas these TPH, they're kind of restricted to the lung. And they express the same sort of signature as these peripheral helper T cells that have been characterized in a number of various inflammatory diseases like rheumatoid arthritis. So we wanted to actually look at what the shared and kind of tissue-specific transcriptional features of these cells are, and what we can see is that TFH in the lymph nodes, they have CXCR5 and BCL6, as we'd expect. This is highlighted by the blue bars on the left side of the graph. The green dots on the right side on the bottom right of the graph, they're characteristic of the tissue-specific features of the peripheral helper T cells with interferon gamma and IL-10. But what's really nice is that they share this core signature of B-cell help, including IL-21, PD-1, CXCL13, in the absence of the CXCR5 and BCL6 that's characteristic of TFH. These cells, the TFH in the lymph node and the TPH in the lung, they're both clonally expanded, so we know they're doing something, we know they're responding to something. But we wanted to ask a kind of interesting question about the clustering, the clonal overlap between these two cells. And what we find is when we look at the clonal overlap of the cells in the lung and the lung lymph node, we see that there's clones that are only found in the lung TFH. Those same clones are found within the lung, and those same clones take on a TPH phenotype, which suggests that they have a common progenitor. But I haven't actually shown any of you guys where these cells tend to localize to. So we turned to some xenium analysis for this, and we have some we have a lung and a lung lymph node here. The blue is the B cells, so we can see a load of follicles in the lung lymph node, and we can actually see a number of bolt structures in the lung there. And what was kind of interesting to see is that these bolt structures on the cellular level have a very similar cellular composition as the lymphoid follicles there. And this is a bit of a complicated graph, but I'll take you through it. Basically, the horizontal axis is distance from bolt. And the left column is the center of bolt, and then the line in the middle is the edge of the bolt. And what we can see is there's a number of genes upregulated in the CD4 T cells themselves that match what we saw in the single cell sequencing. So we can kind of be confident that these are the cells, these TPH-like cells are characteristic of the cells that are helping B cells in bolt, in comparison to the very canonical TFH-like signature in the lymph node. And the kind of final data slide, I've got loads more data upstairs, so we can chat about it after, but we kind of figured out that PD-1 and ICOS was a fairly good surrogate for these TPH cells at the protein level. They express more IL-21, and crucially, these follow the same sort of dynamics throughout childhood as the bolt does. So, as a kind of like summary of how the Michelson's going to help me kind of take this further is to kind of expand this into more organs and figure out the mechanisms for the development of this bolt, further dissect the functional output of the bolt themselves and what the kind of characteristics of these peripheral helper T cells kind of induce different antibody responses in these mucosal sites, and then expand this to multiple tissues. So we want to look at Peyer's patches in the ileum, isolated lymphoid follicles in the jejunum, and then compare them to their mesenteric lymph nodes in paired donors as well. So this kind of paradigm suggests that these bolt structures are very specific in early life, which kind of has fairly obvious implications for the kind of relevance of mucosal vaccine design and how we need to think about when we're going to vaccinate these donors. And that's kind of it, I think. Yeah, and I'd just like to thank my mentor, Donna Farber. You probably could have all guessed that this was kind of her tissue resource that we're kind of using here. And yeah, that's kind of it. I've got a poster upstairs if anyone wants to chat. Please bring a beer.
Thank you. Thanks. We hope everyone took something away from this. And again, all the presenters here will be floating around, so please do find them if you have any questions or want to continue the conversation. The Michelson next round of Michelson Next Gen Grants is also open at the moment, so if folks are interested to learn a bit more, we'll be at the table to field any questions and have conversations as well. But appreciate you all listening through the dinner, and we hope to see you soon. Thank you.